Hi, welcome to this video on incomplete dominance versus Mendelian inheritance. When researchers and scientists repeated Mendel's monohybrid experiments in other plant and animal systems, they found that the law of dominance did not always apply. Instead, the hybrid often showed an intermediate or incomplete phenotype, leading to the emergence of the concept of incomplete dominance. In this session, I will focus on incomplete dominance and compare it with Mendel's law of dominance. So in this slide, I will explain the experiment conducted on 4 o'clock plant that led to the emergence of new concept known as incomplete dominance. And this is the experiment which also denies the law of dominance of Mendelian inheritance. So in this case also in P1 plant they selected pure breed contrasting phenotype for a single trait. In this case color. Pure red flower was crossed with pure white flower. Genotype is given here as capital R1, capital R1, and for white it is capital R2, capital R2. The reason for choosing capital letters I will explain in a minute. So this cross produced in F1 generation all plants bearing pink flowers. This is monohybrid. It does not match, the phenotype did not match the parental phenotype, therefore it does not support law of dominance. It supports law of or incomplete dominance. However, there was a question what happens to the parental phenotype red and white. To address that, they self-crossed these F1 monohybrid, and in F2 generation they found that 25% were pure red, 25% were pure white. So there is segregation of the parental phenotype and they retain their identity. So it supports law of segregation, although law of dominance is not supported by this experiment. And they 50% were pink flower with R1 R2. Again R2 R1 R2 is both in capital letters, and the reason for that is that both R1 and R2 alleles they are producing an intermediate color. So basically F2 generation the phenotypic ratio is 1 is to 2 is to 1, whereas in Mendelian according to Mendelian principle the phenotypic ratio is 3 is to 1, one red, two pink, and one white according to incomplete dominance. So there is a difference in the phenotypic ratio between Mendelian inheritance and incomplete dominance. The genotypic ratio is also same, 1 is to 2 is to 1, 1 homozygous red, 1 homozygous white, and 2 hybrid pink. So in the hybrid both alleles are dominant, that is why the capital letters have been selected, and they produce an incomplete phenotype which I will explain in the next slide. So I will explain here the pink phenotype of the monohybrid. In monohybrid, R1 produces red pigment, R2 produces white pigment, and these individual phenotypes mix and produce pink phenotype, which is intermediate color or incomplete color. So in monohybrid, both alleles are dominant, that is why they are shown in capital letters. However, alleles do not mix or blend, they retain their identity and they segregate in F2 generation. We have seen that. However, the individual phenotypes mix and produce an incomplete phenotype, giving pink color or intermediate color. So in summary, incomplete dominance refers to the hybrid condition. In hybrid, both alleles are dominant. Both alleles produce their corresponding phenotypes. These individual phenotypes mix and produce an incomplete phenotype or intermediate phenotype. However, there is no blending or mixing of the alleles. These alleles, they separate, they retain their identity on transmission. So here is a query. What are the genotypic and phenotypic ratios in the F2 generation in incomplete dominance? The answer is very simple. The phenotypic ratios in this case I had described is 1 is to 2 is to 1, 1 red, 2 pink and 1 white. Genotypic ratio is also same, 1 is to 2 is to 1, 1 homozygous red, 1 homozygous white and 2 are pink. Thank you very much for watching this video. Please like it, share it and subscribe it. I will be uploading more videos on the concept of Mendelian inheritance. So keep your eyes open. Until next time, you take care. Bye.